So continuing on with chapter 3. So the type of microscopy that you will be doing in lab is referred to as bright field microscopy. And this is because the background is going to appear white and then the specimen is going to have color to it. This is by far what's widely used in medical labs, research labs, diagnostic labs. So it's basically you're taking light, you're transmitting it through the specimen, and then looking under the ocular and objective lens to see the image. So you can do this with, organisms that are alive, or you can do it with ones that are dead, meaning preserved and stained. And that's what you guys are going to do. So everything that you're going to look at under your microscope is going to be dead, and then you're going to stain it. An example of how it could be used with a live specimen is, for example, if a person has some type of intestinal parasite like Giardia, the way that med labs diagnose that is they take a stool sample and then they actually have to see the Giardia alive and kind of swimming around to be able to diagnose that. So that's an example where that would be used. But you guys are going to kill everything on your slide and then you're going to stain them. Another type of microscopy, which is the opposite of what you're doing, is called dark field microscopy. And this is where the background looks black or really dark, and then the image or the bacteria would look, white, or have some kind of contrast against that dark background. So this is useful for when you want to look at living cells that, maybe if you were to preserve them and stain them, they might get kind of distorted, or you need to see them alive. But it's used less commonly when we're talking about microbiology. But yeah, bright field and dark field are basically the opposite of each other. So here's an example. This is your garden variety paramecium, which I'm sure you guys looked at in high school biology class. So this is what it would look like under bright field microscopy. As the background is white or light. Here's dark field. And then this guy, this is kind of interesting. This is what is called phase contrast. And in larger medical laboratories, you'll definitely see a microscope like this. It allows you to see external structures, like you can see the little hair-like cilia on the outside. You can see some internal structures. Much more expensive, a much more powerful microscope. But normally in bigger hospital labs, you'll see one of those. And it's predominantly used for looking at live specimens, like I said, an intestinal parasite. Okay, then we have electron microscopy. Now, this is something that we don't have at Kirkwood, and if we did, well, actually, you should be really glad we don't have this because your lab fees would be like $10,000 a semester because electron microscopes basically take up an entire room. They are super expensive to maintain. So, and plus, when we're studying biology in terms of diagnostics or microbiology, we really don't need it. But electron microscopy is really useful because it can magnify an image between 5,000 and 1 million times. So you can see things that are super, super small. Keep in mind, the maximum total power of magnification of your microscope is 1,000. So obviously it's a lot more powerful. So electron microscopy was actually developed for studying, fossils and rocks and, non-living things. But pretty soon biologists were like, hey, we've got little stuff too. Let us look at it. And it allowed the fine structure of cells like organelles to be seen on a molecular level. This is how viruses were discovered because viruses are so incredibly small, you could never see them on the microscope that you guys use. So, the field of virology didn't start until really about the 1950s when viruses were discovered by using the electron microscope. So there are two different kinds of electron microscopy. One is called transmission electron microscopy, abbreviated TEM, and scanning electron microscopy, abbreviated SEM. So this is what transmission electron microscopy looks like. And this is where electrons are blasted through the specimen. And so you basically get a cross section. It's very similar to if you've ever had a CT scan. If you've ever watched the, say the radiologist or your doctor kind of look through the images, basically it's taking slices of your body and putting together an image. This is very similar to what transmission electron microscopy is. It's basically thin slices and a cross section. Okay, so down here, this is a paramecium too, but now we're able to see cross sections with all of these different organelles that are in there. And like I said, viruses were not discovered until electron microscopy was used. And this is actually HIV. That's what HIV looks like. We'll talk much more about that when we get to our unit on viruses. Scanning electron microscopy is different because it isn't a cross section. It shows external structures only. So it's a lot prettier. I mean, it's kind of like art, like this is an algae cell. We don't study them in micro because they're not pathogenic, but they're pretty. They're neat to look at. Here is the paramecium. So you're not seeing external structures, but this is actually, God, I believe this is a stomach biopsy. And so you can see there's bacteria shown here in orange. There's cells from the actual person. And then you have a big old paramecium in here. So transmission is a cross section. The electrons go through the specimen, whereas scanning, you're only looking at external structures. Oh, here's an example. So I was talking about Giardia, which is an intestinal parasite, which, by the way, you can get right here in eastern Iowa. I used to think that Giardia was, that's something you get when you go on, an exotic vacation south of the equator, but nope, it's plentiful here in Iowa's waters. So this shows the Giardia parasite, and Giardia actually sucks onto a person's intestine. And so you can actually see the mark left by the sucker on the Giardia. So kind of a cool image, scanning electron microscopy, but also, kind of creepy when you think of millions of those being in your intestine. So how do we prepare specimens to look at on our microscopes? And this is what you guys are going to start doing in lab 3. So, no big surprise, we're going to mount a bacterial sample on a glass slide. And there's a couple of different ways to do that. Okay, so how we decide to do our preparation is, do we want to look at a living sample or do we want to look at something that's dead and preserved? So in our case, our samples are always going to be dead and they're going to be preserved. And also, it's the goal of whoever is looking in the microscope. Do we want to see things swimming around, or do we just want to look and say, what is there? Is it bacteria? What is the shape? What is the arrangement? What is the gram type, which we'll get to in a little bit? And also the type of microscopy available, which for you guys is the basic light microscope, bright field. Okay, so yeah, we're gonna preserve everything, and we're gonna use it on a bright field light microscope. So the smear technique that we are gonna use was invented by a guy named Robert Koch, and you'll hear about him more in class. He actually did a bunch of other things that were very significant in microbiology, but he was the first one to say, hey, let's take some sample that we think has bacteria in it, put it in a drop of water on a slide, and then look at it. Okay, so we are going to do the same thing, and then we're also going to do what is called fixing the smear using ethanol or alcohol. And, we'll talk about that in a little bit. And of course, I'm going to give you a demo before you do any of this by yourself in lab. And then after we've fixed the smear so that the bacteria stick to the slide, then we're going to stain it. And the staining allows us to bring out the contrast. And so we can see the bacteria. We can see what shape it is. Is it cocci or rods? What arrangement is it in? Is it in staph or strep? and, allow you to see that on high power. So we're going to use some dyes to be able to do that. And when we talk about dyes that are used in microbiology, there are two different types of dyes. Type 1 is called basic, type 2 is acidic. And basic dyes, okay, and this is important, have a positive charge. So all of the dye chemicals have a positive charge. Acidic dyes have a negative charge. And the charge is what determines how it interacts with the bacterial cell. Okay, all of the dyes that you are going to use in lab are basic dyes, okay, that are positively charged. So let me go back to when we're talking about fixing our smear. Okay, so you'll hear me talk about this in lab, and you'll read about it in lab 3. So when I say fix, what that means is we are going to kill the bacteria, and we're going to use this process to make sure that they stick to the slide. And the way we do this is to use alcohol, in this case, ethanol. So when we do the fixation step, that has two purposes. Okay, first of all, ethanol evaporates really quickly and it dries out the cells and kind of shocks them. So what that's doing is it's killing the bacteria. So we don't have to worry about it causing infection anymore, even though it could potentially be a pathogen, because we're going to kill it, first of all. And then also what the ethanol fixation does is it denatures the proteins in the bacterial cell wall so that the bacteria stick to the slide. If we skip the ethanol fixation step, what could happen is you stain your bacteria and you look at it under the microscope and there's nothing there. Well, what has happened is because it wasn't fixed to the slide, it basically rinsed off during the staining process. Okay, so we always need to remember to do the ethanol fixation step. Those are the two purposes of that. So I mentioned positive and basic dyes, okay? And here is why those charges become really important, okay? Every single bacterial cell, and it doesn't matter what species it is, has a net negative charge. And it has to do with that chemical that I mentioned called peptidoglycan, okay? So any bacteria, doesn't matter, what kind, what species, it has a net negative charge. So if we use a basic or positive dye, opposites attract, right? So the positive dye comes here and it's actually sucked into the cell. And so the cell becomes stained and the background remains white. Okay, with negative dyes, it's the opposite. Okay, so a negative dye would come here. Here we have a negative, the dye would be negative, like charges repel. And so in negative dyes, the dye does not enter the cell. Instead, it produces a dark or black background and the cell remains clear or white. Okay, but we want to look at cells that actually have some color to them. And so that's why we use positive dyes, because the negatively charged bacteria will suck the positively charged dye into the cell. And so it will have color. So just to kind of, summarize that. So positive staining, positive dyes. Remember, positive dyes are also called basic dyes. The dye sticks to the specimen to give it color. The dye is actually sucked into the cell because the cell is negative and the dye is positive. In negative staining, the dye doesn't go into the cell. Instead, it kind of settles around the boundaries, creating kind of a silhouette. So some common negative dyes are called Nigrosin, India ink. Nigrosin is a black dye. India ink is very dark blue. Negative staining is very useful when, let's say, for example, you work for the DNR and you have a little field microscope and maybe you want to stop at different points along the Cedar River, take a water sample and just kind of see if there's a bunch of bacteria, cocci and rods. It doesn't require ethanol fixation, so you can take a drop of water, add some nigrosin, and look at it under, a field microscope. It's kind of a quick and dirty way just to say, oh yeah, there's a whole bunch of rods here. I need to take this sample into the lab for further analysis. So let me show you some examples of these. Okay, or let me get through this first. So here's the positive dyes, and you can see by far that's what's most commonly used in microbiology. So the ones that you're going to use in lab are crystal violet, which is purple, and saffronin, which is pink. But there's dyes basically every color of the rainbow. Okay, and the types of stains: simple stain, which you're going to do in lab 3, then we're going to get to a gram stain, and we're actually going to talk about these other ones. Negative staining is kind of, we just want to look and see, is there bacteria there and what shape? Boom, that's about it. Okay, so here's an example of positive stain. So here we have some cocci, and this is stained with crystal violet, which is purple. This is stained with saffronin, which is pink. Both of these dyes are chemically the same. They work exactly the same. They're both positive dyes. They just happen to be different colors. And when in microbiology, under the microscope, these are going to be the only two colors that you're going to see. So your bacteria is either going to stain purple or it's going to stain pink. Here's an example of negative staining. So yeah, it doesn't really, it can say, oh, I have bacteria, looks like cocci, and it looks like it's in a staph configuration, and that's it. So yeah, it's really useful out in the field, looking at water samples, and it gives the person just enough information to say, ooh, that's a lot of bacteria. I'm kind of concerned about water quality. I'm going to take a sample back to the lab and do further analysis on it. Okay, so I'm going to stop there and get this uploaded, and then we'll pick up with simple stains. Thank you.